First, I'd like to thank the organizer for having invited me to this conference, and especially to Kelly Michaels, who has really helped me to set it up. I will talk today about PCSK7, which is a novel regulator of apolipoprotein B, and we believe is a potential target against metabolic dysfunction-associated steatotic liver disease. The interest in the laboratory for many years has been to understand how secretory proteins actually enter the secretory pathway and get activated by various post-translational modifications. The most irreversible one is known as the cleavage site that transforms a protein from an inactive form, like just proinsulin, for example, into an active insulin. But it can also result in an inactivation. And so for many years, we try to understand the members of the family of proteins that we identified that are implicated in these processes. And we've identified a family of 9 members, known as the proprotein convertase family, and their names vary from PCSK1 to PCSK9 in the gene, as well as the enzymes themselves. They can have names like PC1, PC2, PC7, and so on. The first 7 members of the family cleave secretory proteins at specific basic residues, as you can see here by the motif of RXKR. They are serine proteases related to bacterial subtilizin's. They have the aspartic histidine and the serine of the active site. The other 2 members, SKI1 and PCSK9, are also members of the family, except they don't cleave at basic residues, but rather they cleave at non-basic residues. Today, I will speak to you about 2 members, PCSK7 and PCSK9, and how these 2 members in a non-enzymatic fashion can affect steatosis in the liver. Now, these 2 enzymes are implicated in various diseases, such as cardiovascular diseases, pathogenic infections, including SARS-CoV-2, HIV, and others, as well as in cancer. So the first member of the family that was identified to act non-enzymatically, but to regulate LDL cholesterol, has been PCSK9 in 2003 and shown to be a unique convertase. Indeed, when we first studied the synthesis of this protein, we realized that it is synthesized as a precursor protein, as always, as a proprotein that is autocatalytically cleaved to release the functional active enzymes, but it is also released unexpectedly with its inhibitory prodomain that remains strongly stuck with the enzyme, suggesting that this protein will no longer act as an enzyme. What we next observed is that it is also secreted with a fragment known as, which we have identified at the cleavage site to be RFHR, and that is a furin cleavage site. And that results in an inactivation of PCSK9, you will see later, and that the product that is resulting from that, which is coming now from 219 till the end, turned out to be a gain of function on oxidized LDL. Now, what is this protein doing? It's extremely rich in the liver, as you can see, and in the small intestine. And this reminded us, when we saw this, of the patients that we see in our clinic, which develop what is known as xanthoma and xanthelasma. In fact, Leonardo da Vinci has observed in his model of Mona Lisa that she had xanthelasma. I didn't know what that means at that time, the 15th century, but she died at the age of 37. So the question was asked, could PCSK7, richness in the liver and on chromosome 1, P32, be related to cholesterol metabolism? That was confirmed when we collaborated with 2 fantastic groups, one there in Paris, and they were interested in the problem of hypercholesterolemia. And Catherine Boileau was the leader of this group, and Mathilde Faré, as well as Mariana Bifadel. Together, we discovered that mutations in this gene called PCSK9, single point mutations, phenylalanine 216 to leucine or serine 127 to arginine, are directly related to hypercholesterolemia, and that all patients had about 1.5 to 3-fold higher LDL cholesterol. Once this was published, a number of countries have identified also mutations in PCSK9 that result in what is known as a gain of functions, and that enhances the cholesterol levels, and also other mutations that result in a loss of function, whereupon the cholesterol is about 1.5 to 4-fold lower. In fact, 4 people in the world do not have PCSK9 at all, okay? And these people live very well, but their cholesterol is 8-fold lower than you and I. 8-fold. That is very high. This, of course, led to us to identify the mechanism by which PCSK9 could regulate the LDL cholesterol. It turned out that it binds the LDL receptor and drags it to lysosomes for degradation, as you can see here. And so an inhibitor of PCSK9 would, of course, increase the number of LDL receptor at the cell surface and mop up the excess LDL cholesterol circulating, and that's why it's hypocholesterolemic. However, how does the LDL receptor go to the lysosomes when it's bound to PCSK9? We recently solved this problem and found that it requires an interaction with one of the members of the MHC class 1, and that directs the whole complex to lysosomes. So this is how it looks like. If you take a human hepatocytes and incubate it with a PCSK9 monoclonal antibody, it lights up like a Christmas tree, as you can see. The LDL receptor levels go very high, as well as the internalized LDL into the liver. So, of course, this led a number of pharmaceutical companies to try to develop different inhibitors, and there are many of them, from gene editing to antibodies, to siRNA, even to vaccines, as we're now going, and in small molecules. But the only two that are really accepted so far by the FDA are the monoclonal antibody and the siRNA. What does this have to do with what we're going to talk about today? And that is hepatic liver disease. Well, it came out from the fact that when we took our mice, knockout mice, which survived very well, and treated them with a high-fat diet, high cholesterol, high fructose, high sucrose, high lipid levels, and what we found is that they have accumulation of lipids in the liver. That was not good, because we can remove the cholesterol, yes, by the absence of PCSK9, but we increase the lipids in the triglycerides in the liver. And this has to do with the fact that PCSK9 targets another protein called CD36, which is a transporter of the lipids. And in fact, absence of PCSK9 increases a lot the amount of CD36 at the cell surface. But amazingly, if you do exactly the same experiment, except instead of giving a knockout, you just give the evolocumab, which is the antibodies that blocks the circulating PCSK9 ability to bind the LDLR, you do not get an increase in CD36. So the conclusion came that antibodies and knockouts do not give you the same phenotype. And we came up with the model that indeed the PCSK9 binds the LDL receptor by its catalytic subunits and directs it to lysosomes. And if you block it with the antibody, you increase the level of LDL receptor. But we don't touch CD36 because the CD36 binds rather the C-terminal domain of PCSK9, and that's what sends it to lysosomes. So even though it's still bound to the antibody, it's active in degrading CD36. So how is that useful for mast cell? It turned out that a number of patients now have been treated with a monoclonal antibody to PCSK9. And those that had computer tomography, you can see here that they had a homogeneously hypodense liver, and the portal and hepatic veins, there were unclear delineation. But when we treated them with what was treated with a monoclonal antibody, clearly see now that the liver have increased density and the portal and hepatic veins to be clear. This suggests that we reversed the phenotype. In another set of patients, the monoclonal antibody showed a dramatic response in this patient who failed conventional therapies. And the encouraging results seem to merit research into use of PCSK9 inhibitors, such as monoclonal antibodies, as a first-line therapy for the acute phase of NASH. What we propose is that you should use the monoclonal antibody rather than the RNA approach for the treatment of mast cells and MASH. Now, this leads us to PCSK7, which is the seventh member of the family. This is a membrane-bound protein that was for many years thought to act as an enzyme as the other members, the other 6 members above it. But we have found very recently that in mammals, this enzyme mostly acts non-enzymatically. And the first non-enzymatic identification was its ability to bind apolipoprotein B and act as a chaperone and allow it to exit the endoplasmic reticulum from the liver. And so we published this work a few months ago. Vatsal Sashin was the first author and showed that PC7 is a regulator of ApoB and a potential target against non-alcoholic fatty liver disease. How did we reach that conclusion? Well, we first did epidemiological studies in human and shown that gain-of-function SNPs, such as here in intron 9, are associated with increased triglycerides and ApoB in circulation. That means a lot of PCSK7 is associated with a bad phenotype, which is triglyceride and ApoB that is high. On the reverse side, if you have a loss of function, this is a case of a single point mutation of a protein to a leucine in the cytosolic tail of PC7, that results in the reverse phenotype that is lower triglycerides as well as lower ApoB. Now, how does that work? Well, we had to recur to knockout mice of PCSK7 who live very well, by the way, like PCSK9. And we call them the cool mice because they have lost a lot of their anxiety. It has to do with BDNF in the brain. But basically what we see is that in the mice, when we give them a normal diet, that in the liver as well as in the plasma, about 50% lower levels of ApoB. So looks like PC7, absence of PC7 decreases the level of ApoB that gets out of the liver. What is the effect of that? Well, we think that has a direct effect that there is much less accumulation of triglycerides in the liver. How can that be? If you diminish ApoB, why would triglycerides in the liver diminish? Well, we think it has to do with what happens to the triglyceride, what happens to lipid droplets. If you give the animals a high-fat diet, okay, then the absence of PC7 no longer has any effect. So it has to do with the ability of PC7 to reduce the lipids when there is no high-fat diet. So we have gone into the mechanisms of that and found that this is applicable in human as well as in mouse, in rats and mice. And basically that the ApoB is decreased 50% even in human hepatocytes. The mechanism, we believe, has to do with the fact that if you do not have PCSK7 very early on, within the first few minutes of synthesis, ApoB gets degraded. Where does it get degraded? It gets degraded, we believe, in the proteasome because lactocystine and MG132 block this effect. The most interesting point here is that this effect of PCSK7 is independent of its enzymatic activity, because if you block the enzymatic activity with a specific inhibitor, it still works very well in allowing ApoB to be secreted from the liver. So the model we came up with is that PC7 is a chaperone of this long protein ApoB. As it gets translated into the ER, it allows it to fold. In the absence of PC7, it results in an unfolded ApoB, which can be degraded by the proteasome, and that leads to accumulation because then no longer VLDL can exit the ER because there's much less ApoB. And so it accumulates in the ER, causes a UPR activation, and we believe that results in autophagy and beta oxidation, and that's why we have less lipid accumulation. The most important point here is that this UPR activation is a functional UPR activation that does not result in ER stress at all. So can we exploit this? So what we said was that maybe because of the fact that in the high-fat diet, the mice will still develop steatosis, can we give them a high-fat diet for a while and then let them recover on a normal diet? Will they be able to recover? Will they be able, in the absence of PC7, once you get the steatosis, to recover? And the answer is yes. They recovered very well about 4 weeks after the treatment. So the idea comes, what do we do with that? How can we move this to clinic? And so Linus Pauling said that the best way to have a really good idea is to have lots of ideas. Maybe one of them would work. And so we're asking ourselves the question, can we reverse the diet-induced metabolic dysfunction-associated fatty liver disease if we give them an antisense oligonucleotide or an siRNA against PCSK7? And so we were inspired by the multiple works that were done with antisense oligonucleotide for the treatment of various diseases, especially those that target the liver, and where they use N-acetylgalactosamine attached to the oligo, and that allows them to be targeted specifically to the liver because of a receptor known as SGPR that is only expressed in hepatocytes. That allows the antisense oligonucleotide to come in, and it degrades the RNA. Indeed, we use this technique. We took animals, gave them a high-fat, high-cholesterol, high-fructose diet. They developed steatosis, and then gave them a period of 4 weeks, 8 weeks to recover from that onslaught in the presence of one of the two best oligo that we have identified, ASO2 and ASO7, coupled with the trientennary N-acetylgalactosamine. And so this is how it looks like. We injected 2 injections per week during the 8-week recovery. What did we find? Wow, look at what we found. We found that indeed both ASO2 and especially ASO7 result in very, very significant reduction in lipid droplet accumulation in the liver after the recovery period. This was associated with a very nice recovery of the liver that no longer accumulates fat, much less liver inflammation, much less hepatocyte ballooning, and much less steatosis. Not only that, we also found a significantly reduced fibrosis, which these are the hallmarks of NASH. And this was associated indeed with a 50% at least decrease in ApoB, as we saw in the knockout. So the conclusion that we've reached is both from human, from cell biology, from mice, and now treated mice with the oligonucleotide, that maybe this indeed is a treatment for non-alcoholic fatty liver disease. This non-alcoholic fatty liver disease is a major disease that affects our society, and it can result in 20% that develop into cirrhosis, and about 4 to 27% that will develop hepatocellular carcinoma. So it is a deadly disease, and we really have to treat it early. It is a cumulative disease over age. We believe that an ASO treatment, before it reaches the cirrhosis stage, can be used to reverse the MASH treatment, and will that PC7 be a pharmacological target? So what is mast cells? It is often associated with metabolic abnormalities, including obesity, insulin resistance, type 2 diabetes, hypertriglyceridemia, and dyslipidemia. Now, this is due to genetic predisposition, abnormal lipid metabolism, oxidative stress, inflammation, lipid toxicity, mitochondrial dysfunction, gut dysbiosis, endoplasmic reticulum stress. Many, many factors are implicated in mast cells. So we believe that because of this numerous metabolic and pathological alterations that proceed in parallel, no single action of a drug will work. In fact, there are about 300 different clinical trials going on around the world for different targets to be able to use in this disease. The 2 best targets that so far are working on the NASH landscape are thyroid receptor beta agonists and glucagon-like peptide 1 receptor agonist known as semaglutide, or Ozempic if you want, as we hear about it in the news. What is the functional result of treatment of people with a thyroid receptor beta agonist? This was just accepted by the FBI. If you want a clinical trial, phase 2, that shows that about 16 to 20% of the patients do respond to this resmetirom drug, an orally active drug that inhibits thyroid, that activates thyroid receptor beta, but only about 15 to 20% reduction in the phenotype has been observed. So there's still about a large amount of people who do not respond to this drug, and the response is relatively mitigated. Not unexpected, because this is a very complex disease. So there is space of combining this drug with another approach. So these are our conclusions. We believe that silencing of PCSK9 enhances the degradation of cell surface proteins such as the LDL receptor, VLDLR, and CD36. That PCSK7 is a molecular chaperone, escort of specific secretory proteins of ApoB and like other, and that we believe it would be useful to try to silence PCSK7 with an ASO targeted to liver and PCSK9 circulating with a monoclonal antibody, and together they may synergize anti-mast cell treatment. So I'd like to thank all the members of our group that really resulted in this monumental work, especially Vatsal Sashan, who was driving this PCSK7 study. Thank you very much, and I'm open to questions if you wish.